5 major biological types, and those are protozoa, algae, viruses, microscopic fungi, which includes molds and yeast, and then bacteria. So this list, that is protozoa, algae, viruses, microscopic fungi, and bacteria, this classification is on the basis of or according to the structure of the microorganisms. But it sometimes become more convenient, particularly when we are talking about food microorganisms in relation to their role with the human life. Okay, how they influence our lives, our food system, our health. So if we classify microorganisms keeping that view in mind, like in relation to their role in human lives, microorganisms can be classified into 4 major categories like pathogens, spoilage organisms, beneficial organisms, and inert organisms. Okay. Although it is convenient to use a functional classification, but it may be emphasized that it is always not a hard and fast division, because an individual organism in differing circumstances may fall into each of these categories. Okay? Each of the 4 groups like pathogens, spoilage, beneficial, as well as inert organism, there are certain microorganisms which are at times beneficial, at times the same microorganisms are not desirable, not beneficial. Even a single bacteria, single microorganism sometimes under different circumstances can be classified and can perform the role of a pathogen. It can perform the role of a spoilage organism, beneficial organism, or inert organism. So the best example in this case is the E. coli. E. coli, it is generally considered to be an inert organism. However, in some cases, it may be pathogenic because there are certain strains of E. coli which cause diseases. Okay? There are certain strains of E. coli known in the literature which spoil the foods without causing any illness, without producing any toxic. They just spoil it, so therefore they become spoilage. Also, there are certain strains of E. coli which are used for beneficial purposes, particularly in the eggs, etc. Before the drying of egg, this E. coli is inoculated with the egg white and egg yolk, where it removes the glucose. So E. coli, that is enzymes, glucose oxidase enzymes, etc., contained in it, it converts the glucose as this bacteria utilizes the glucose or removes the glucose, you can say in other words, from egg white, and therefore it becomes a beneficial microorganism as far as the processing, because otherwise glucose may cause browning of the egg powder. So here means that is the same microorganism under the different circumstances is categorized into all the groups. Okay. So regarding structural classification, I think protozoa, algae, viruses, microscopic fungi, etc. molds. So protozoa, they are small single-celled animals. Okay. They are motile and live in water or ponds. So the algae are simple plants. Some of the algae are microscopic like seaweeds, etc. But many are microscopic. Algae generally they are plants, so they contain chlorophyll or similar pigments which enables them to photosynthesize. right, do photosynthesis. Okay. Some algae like chlorella, etc., they are grown on the surface of water. They are harvested as well as they are used for use as a source of protein. So some algae are very good sources of protein also. Okay. Viruses, you are all aware, I hope, that is the COVID virus, you know, that they are they are the smallest of all microorganisms, varying in size even from 30 to 300 nanometers. Okay. They are usually acellular, that they are generally acellular and made up of a central core of nucleic acid, which is surrounded by a protein coat. And they always live as a parasite. That is, they depend on larger living cells, etc. Then comes the category of microscopic fungi. These fungi, they are also plant, but unlike the algae, they do not possess chlorophyll, and therefore they are unable to photosynthesize, and they require complex organic compounds as food. Okay? These microscopic fungi, they may be parasites or saprophytes. That is, those which take food, which depend, grow and feed on dead organic materials are termed saprophytes, and those fungi who depends or take their food from living plants and animals are termed parasites. Okay. So in this microscopic fungi, molds and yeasts are two major classes, categories, and the molds are usually multicellular. Each mold growth consists of more than one cell. However, each cell is capable of independent growth. And molds, normally they reproduce by asexual process. That they reproduce by asexual spores by fusion of the hyphae. They contain hyphae in there. Okay, and this molds, certain molds are not desirable. They spoil foods, etc. Okay, they are particularly in the case of refrigerated foods, but there are certain molds which are used for beneficial purposes. Certain molds are used in cheese making, like Danish blue cheese, Roquefort cheese, Camembert cheese, or even there are many mold ripened cheese. Okay, molds are also used in the production of soya sauce. Some products used in the food industry like citric acid, etc. Okay. Also, some molds are used for producing antibiotic like penicillin. Okay. Then comes the bacteria. Sorry, before bacteria, we'll go to the yeast is remaining. Yes, these are also important from the food. In the food microbiology, they are simple single-celled fungi. They are mainly saprophytic and usually grow on plant foods. And these yeasts normally they reproduce sexually by a simple process known as budding. So in the budding, what that is the cytoplasm in the cell at a particular location is bulges out, and then it grows. Nuclear materials is formed. Then it particularly the bulge, it detaches itself. It removes itself from the parent cell. So that's how this mold is multiplied. That's how mold multiplies. Okay, and the economic importance of the economic importance of yeasts lies in its ability to break down carbohydrate food into alcohol and carbon dioxide. That is very, very important reaction which these yeasts they catalyze, they encourage, they participate in it. Like the glucose is converted into ethyl alcohol and carbon dioxide by this yeast. There is a yeast Saccharomyces cerevisiae, which is known as a bread yeast or also wine yeast. So this is the Saccharomyces cerevisiae. It contains certain enzymes which is called gymase enzyme. And this gymase enzyme, it actually converts sugar into alcohol and carbon dioxide. And this is the major basis of the manufacture of that most of the alcoholic beverages. This is the reaction that is the Saccharomyces cerevisiae catalyzed that conversion of sugar into alcohol and carbon dioxide, which is in the products like fermented bakery products like bread, etc. This is the major reaction. Okay. So in this case, that these yeasts become very, very important, and for that reason, these yeasts in the market, these are harvested. These yeasts are cultivated commercially to be sold as a bread yeast or wine yeast for the bakery industry or wine industry, etc. But for the commercial production of yeast, they require plentiful oxygen. So in the plentiful oxygen, where there is a sufficient supply of oxygen, then there is a complete breakdown. Like this sugar, glucose C6H12O6, it takes 6 oxygen and gives 6 moles of carbon dioxide and 6 molecules of water. So upon complete oxidation of glucose, there is carbon dioxide, water, a lot of energy is liberated. So here, in this case, that is the important thing is that when the same yeast, when it grows in the wine industry or bakery industry, mostly anaerobic conditions are required. They favor that alcoholic fermentation. Okay, in the absence, that is alcoholic fermentation of glucose by yeast takes place in the absence of oxygen, whereas complete oxidation, production of carbon dioxide, water, and energy, it is a aerobic process. So when the yeasts are grown commercially in the field for making use in the industry, by bakery industry, wine industry, etc. industry, there this yeast is a complete aerobic environment is given. Okay, so this is a brief about this protozoa algae viruses, microscopic fungi. Then we come to the bacteria, and the bacteria, it is a very, very important group of microorganisms. And from food, in the food microbiology, in fact, these are the, you can say that fungi, molds and yeast, algae, and bacteria. These are the type of microorganisms which are of importance, which one must know, one must understand. That is, what are the characteristics, types of these microorganisms, what are the different conditions which they require to grow and multiply, how they can be eliminated from the food, because that is what is done. Okay, so the bacteria, they are very important group of microorganisms because of both their harmful as well as beneficial effects. There are many bacteria which are spoilage. There are bacteria which are infectious bacteria, they cause infection. There are bacteria which cause intoxication of the food. So the food poisoning bacteria, food infection bacteria, or contaminating bacteria, spoilage causing bacteria. So different types of bacteria are there, and they are widely distributed in nature. They are found everywhere: air, water, soil, and moist linings of the mouth, our nose, throat, on the surface of the body. and on the plants. Okay? The bacteria are a smallest single-celled organism, even some being only maybe 0.4 micron in diameter. So small. So bacterial cell, that is basically it contains some liquid or fluid, okay, which is called that is the in the cytoplasm, okay, where various biomolecules etc. are present there in the liquid, and it contains some nuclear material. And this cytoplasm and nuclear material, they are enclosed in some sort of a membrane called the cell membrane, and this whole cell, that is this even above cell membrane, there is a further layer of that is polysaccharides etc. Okay, mucopolysaccharides, which gives a little strength protection to the cell and cell membrane. So that is called cell wall. So means that inside that is a whole. It is a maybe bacterial cell. It may be of different shape, etc. All right, but it is some sort of all these material, nuclear material, cytoplasm. They are enclosed in a membrane and encased in a membrane, and which is further protected by cell wall. And some bacteria, they are provided that even another protection layer, outer layer, that's called capsule. Some bacteria are having flagella, which help bacteria to move, propel in the air, move. Okay, so according this bacterial cell may be of a different cell, like it may be cocci, that is coccus are spherical in shape. Some bacteria which are known as bacillus, they are rod shaped. Some are vibrio, they are usually short curved rods, and some bacteria that they are spirali or spirillum. They are long coiled threads. Okay. Then means that is when the bacteria, they are grown in a medium. Sometimes they may group together, they may club together each other, and that's why there are like cocci or bacilli, streptococci, okay, or that they may be staphylococci. Streptococci means when the grouping is in the form of a chain-like association. Several cells, there are suppose 50 cells, 100 cells, they just attach to each other in a form of a chain. So that becomes a streptococci, streptobacilli. When this association in the form of a clubbing like cluster, or you can say just like grapes are there. So that type of clustering or grouping of the cells is known as staphylococci. Okay. So this is about general characteristics of the bacteria. Then regarding reproduction, the bacteria normally reproduce by a process known as binary fission. Binary fission means that is the just simply nuclear material divided into two half, cytoplasm divides, and ultimately cell wall, cell wall, cell membrane, all the things divide, and each parent cell that gives birth to two daughter cell. Means whole cell equally divides into two daughter cell, and the process goes on. That is the called as a binary fission. Okay. There is another important, very important property of bacteria, okay, related to the food processing, that some bacteria, they have the property of forming spores. That is spore forming bacteria. Means when these bacterial cells are exposed to certain hard situations, some resistant conditions like temperature, pressure, etc. Like every bacteria, we will see little later, it needs some optimum conditions of temperature, pressure, nutrient, and other things for its proper growth, balanced growth. But when these optimum conditions are not available, rather when it is put to some hard conditions, which normally we do in the processing, Then certain bacteria, they convert themselves into some sort of a hard resistant body, which is called spores. So what happens? That is the genetic material, nuclear material, it squeezes itself within the cell into a very small, tiny point-like things, and the cell wall disintegrates, and the cell membrane disintegrate, cytoplasm oozes out. So, but the living part or the component of the cellular component, genetic material, is there in the form of it remains there in the form of a hard resistant body. And during this transition from the living cell to the spores, vegetative cells to spore, there are two important factors. That is, there is a massive uptake by the spore, that is, there is a calcium ion, that is, it is high in the calcium ion. Also, there is some formation of dipicolinic acid. So these two factors, presence of more calcium ion as well as presence of more dipicolinic acid in the spore, make them resistant. So it becomes, the spores become difficult to kill, difficult to destroy. They need more heat, they need more energy for their destruction. So otherwise, they survive, these spores survive the adverse conditions, and when again they get favorable conditions, they grow and multiply. And that is what in many, when we study the food processing technology, etc., you will see in many conditions, these bacteria, when they are exposed to various adverse conditions during processing, handling, storage, etc., then they convert themselves into spore, and they try to survive, they try to resist, but when that's what happens in the low temperature storage, when you put the food into a refrigerator, okay, then they go under the dormant stage, and the bacteria will not be killed, rather bacteria will remain there. But when you take it out of the refrigerator, it will again grow and multiply. So that is the spores formation. But so when we go further, some killing of the bacteria, etc. So normally all calculations, etc., are done on the basis of destruction of spores, not on the basis of the destruction of their live cells. So these are the important considerations about the bacteria. And
Excuse me, sir.
yes, please, yes.
Sir, are you changing the slides?
No, I am just still on the first slide because
Oh, okay,
I'm talking about, you see, that the bacterial cell and their nature, etc.
okay, sir, okay.
Right? So that is just I am giving you because this information, and in fact, I think here just I wanted there, let it be one assignment. You have been already given one assignment, which will be, I think that is one more additional assignment. Okay, Siddharth. Yeah.
Yes, sir, it has given, sir. It has given.
That we are given, but now you upload another assignment. All right. That is, let them give the list of the important microorganisms associated with food in the three categories, that bacteria, yeast, mold, and algae. All right. And both in the case of like they can categorize this, that is a pathogens, beneficial, inert, spoilage. So the list of microorganisms, major microorganisms associated with the food and their characteristics. Let them, that is what is their cell type, how they grow, what are the conditions, etc. So their characteristics are morphology. Okay, so before I come to the growth, there is another very important aspect which one should you should understand, which you should know about the bacteria, because some bacteria produce toxin in the food. Okay, so they are called toxicogenic bacteria, toxicogenic microorganism. Okay, so these bacteria, whatever associated with food, in fact, they can be broadly again classified into two groups. Like one is the food poisoning bacteria, other is the food spoilage bacteria. So food poisoning bacteria may be food infection and food intoxication. And food spoilage maybe that they will just spoil the food, but they may not be producing any toxin. So when we talk about infection, infection means that this bacteria just contaminates, that is it uses food and water as a carrier material. So it just contaminates the food. It may not grow on the food. So food remains good, but the bacteria food is contaminated with the harmful bacteria, and when you consume that bacteria, food contaminated with harmful bacteria, so bacteria along with the food goes into your system in the large intestine, small intestine, wherever it finds favorable conditions, it establishes itself, grows, produces toxin, and then you fall ill. That is what. And this toxin produced by the bacteria may be endotoxin, exotoxin. There are certain bacteria which produce toxin, but toxin remains within its cell. It doesn't releases that cells in the food, that toxin in the food. So that is called endotoxin. So again, if that bacteria cell, it has toxin, but the food is not toxic, food is normal. So you are having the bacteria which has contaminated the food, and bacteria is containing, bacterial cell is containing the toxin. So when you consume such type of food, so it goes inside our system where the cell integrates, toxin is released into our system. So we get disease. Okay. So there are the toxin producing or infection producing, but certain when spoilage causing bacteria, that is what happens. They do not produce any toxin, but rather when they grow and multiply in the food, they consume the food nutrients for their own growth and survival, and in the process, they change the food molecule, food nutrients. They produce secondary metabolites like aldehyde, ketones, carbonyl compounds, etc., and which may change color, flavor, nutritive value, and other aspect of the other quality of the food, organoleptic and nutritional quality. But it is not that the food is spoiled, but it may not be toxic. It may not be disease causing. Like for example, you see that when bread or humid food, bread, when it is left in the humid atmosphere, high moisture food, bread, etc., left in the humid atmosphere, then you might have observed several times the growth of white, cottony growth of mycelium, that is the mold. But that mold is not pathogenic. It doesn't produce, doesn't cause any disease. So if you just trim out that mold and heat the bread, you can eat it. There will not be, but I am not advising you to eat. Okay, but if you eat that, heaven will not fall. Okay, so these are the thing. Now I will come to the microbial growth. So now, yes, please.
Sir, so like you told about food spoiling bacteria and food intoxicating bacteria. So, sir, this food spoiling bacteria, so they don't have any effect on the human health, like the bacterial sheet as well. Like you told us to remove the mycelium
Yes.
growth.
Yes. You see,
So
yeah, I got it. I got your point. There is the one is the spoilage bacteria, other is the poisoning bacteria, intoxicating bacteria, or disease producing bacteria in simple words you can say. So spoilage bacteria, all right, they normally do not cause any effect on the health. They just spoil the food.
okay.
You get
But
that?
sir,
You see,
but sir,
I'm telling. Yes, tell what you tell, please.
but sir, that spoil, they're spoiling the food, right? So that is also intoxicating. Like,
No. That's what we are. That थोड़ा सा उसको clarify कर लो, है ना? कि what is happening if it has that converted the food nutrients into a toxin, then it becomes a poisonous. Food becomes poisonous.
Okay.
There is, all right, because the microorganism, when it grows in the food, what happens? These bacteria, they also need the same nutrients which we need for our growth and survival. So when any food grows, so the bacteria find a medium for it, so they will grow and multiply. So when they are growing, so some bacteria, they have the gut capability that they change these nutrients, normal nutrients of the food into a toxic component. They
Okay.
release some toxic metabolites. Did you get my point?
Okay. Okay.
And
So,
some,
sir,
yes.
so, sir, these food spoiling bacteria, if we eat them, then they will not cause harm. But so they interact with the food and change the food into I mean, change the chemical composition of the food, which makes the food toxic to it. That is why after we eat spoiled food, we face disease. But if we
No,
remove the
no, no. Again, there is a little. I will little clarification is needed. Means that is, if you eat a spoiled food, you may not able to eat. When we talk about spoiled, means the food has changed in its color, flavor, texture, sensory characteristics, maybe nutritional value. All right,
yes,
but
yes.
it is not containing any uh, poisonous material or health hazard component.
Okay.
So that is only referred to as spoilage.
Okay.
But if the same bacteria has grown and it has changed the nutrient into a toxic compounds, or it has produced a toxic metabolites, so that food, it becomes poison.
Okay. Okay.
All right. So you have to make a difference. So that is what, although there are many several, that is sometimes it is just a convenient way has been categorized to make the people understand. But that's why I told you you should not eat a spoiled food, because we never know. Nobody can say with 100% authority until unless you thoroughly examine, evaluate that food for various possibilities, for various metabolites, etc. Nobody can say. But in general, it is said that the spoilage causing bacteria, they change the nutrients into, that is the acid, thiols, or even sometimes they grow and visible presence of microorganisms, etc. may be seen, but they don't, they do not create any health hazard. All right.
Yes, sir. And sir, one more question, sir, regarding that endotoxin and exotoxin,
Okay.
sir. So when we like, sir, endotoxin is only reacts with our body when we ingest it and it gets digested into
Yes.
our system, right? And sir, then exotoxin already like exotoxin, or we can detect the exotoxin contamination from the food itself, like.
Pardon. Can you tell once again?
Yeah, yes, sir. So, sir, like endotoxin, though we can only feel the effect only after we ingest the food because it is contained inside the cell. But, sir, exotoxin, though it is present in the food itself before eating. So,
Yes.
we can draw distinction from the food itself, right? That it contains some toxin.
But what happens? These toxins are so in minute amount, small quantity. All right, that until
Okay.
unless you have a very sophisticated and high-end equipment, and where you could go for a GCMS and MS, etc., such type of, where you can measure the small quantity of the equipment and quantify it and evaluate it, you can do it. Otherwise, but in general, what happens, that is because of their small, the sensory characteristics, appearance, food, etc., that is not changed at all. Means by normal, until unless you go and analyze in a laboratory with high-end equipment, high quality equipment, you will not be able to judge that this food is containing poison or not, toxin produced by bacteria or not. That is
Okay,
the
sir.
reason. That is the reason why people eat poisonous food. You see, many times, several daily, almost we listen some this. So many people died of food poisoning. So many people they consumed this type of poisonous food and like that. Okay,
Yes, sir, got it.
fine. So these are things. Now, should I move further?
Yes, sir.
Okay, all right. So now let's see. So I just gave you a brief of physiology and characteristics of the bacteria, yeast, mold, and fungi or algae. And I would like you to at least prepare some notes, etc. on this, understand the process, because that becomes very, very important in the food. What will it grow, when it will grow, and how it will grow, when it grows, whether it will spoil only the food or it will produce some toxin also. These things, because this information is needed when you preserve the food, when you process the food. So now let's see another very, very important aspect of the microbial growth, because this growth, knowledge about the growth, there are many cases in the food industry where you can almost all fermented food industry, okay, where the dairy industry, fermented dairy products, etc., where the microorganisms are allowed to grow under controlled condition to bring about the desirable changes in the food, in its color, flavor, nutritional value, etc. You can see the very simple example, that is how milk is converted into curd, yogurt, or other products. Okay, so at the same time, many a time we want that the food should, when it is processed, that is with a aim to create condition in such a way that the microorganism doesn't grow in it, so that there is no spoilage of the food or no poisoning, etc. of the food. So how to do that? So for that, you need to understand how it grows, how it multiplies, what are the different environmental factors, other conditions which favors the growth of the bacteria or microorganism. Okay, so now that how do we define growth? Growth is the, that is in a, like in our system, human being or such other cattle, etc. There are animals or living system, even plant also. When there is a growth, means that there is a, you can see by your naked eye visible, that is there is a expansion of the cellular component, their body grows, how the baby is born, and then in the young age. And but in the case of microorganism, always it may not be that by naked eye, by eyes one cannot see that microorganism remains the same like in its cellular composition there, but it may increase in the number, it may even increase in any particular component of the cell. So as far as the growth of the microorganism is concerned, that implies that all chemical components of the cell increases with the same speed, and after a certain time this leads to increase in the cell number, which causes increase in its size or number of the individual or so on. Okay. So in the case of microbial growth, it may be just the process of the orderly increase of all cellular components. So growth is normally performed batch-wise or continuously, and most bacteria have asexual growth, which means that no sex cells are involved in its growth. The bacteria divide, as I told you, binarily, usually perpendicularly to the length axis, and thereby two new daughter cells are produced. So that is the bacterial growth. So mathematical expression of the growth, if we say, so it's clear. But another one more thing, that in any biological system, as I told you, growth can be defined as the orderly increase of all cellular component, as I told you. So in a medium, that is for bacteria, very important is that in an adequate medium to which they are fully adapted, that is when the medium for their growth is perfect, optimum, they which contains all the nutrients which they want, all the conditions are favorable. So in that case, the bacteria are in a state of balanced growth. Is it clear? That in any medium to which they are fully adapted, the bacteria are in the state of a balanced growth. Balanced growth means during the period of a balanced growth, a doubling of the biomass is accompanied by a doubling of all other measurable properties of the population, like protein, DNA, RNA, number of the cell, mass of the cell, or any component of the cell will double. So that is the called balanced growth. The doubling of the biomass is accompanied by doubling of all other measurable components. All right. So if the growth fits the definition that all chemical components of the cell increases with the same speed, a unicellular bacterium increases in cell number exponentially with base 2. So a mathematical expression of the bacterial growth can be given as Nt is equal to N0 into 2 to the power 2 to the power N, where Nt is the cell number at time t, N0 is the starting number at time t is equal to 0, and N is the small n is the number of doubling generations. Is it clear to you all? Yes? Can someone respond?
Sir, can you explain that equation once again?
You see that Nt is equal to N0 into 2 to the power N. Here it is the balance growth. What I told that the in a medium when the bacteria is fully adapted, it is in the state of a balanced growth. Balanced growth means that is the doubling of biomass is accompanied by doubling of all components of the cell. So if the initial number at the time t is the Nt, all right, N0 is the initial number. So after time t, okay, the cell number will be Nt, okay, multiplied by 2 to the power n, where n is the number of doubling, whether it has doubled 2 times, 4 times, 8 times, 16 times, like that. Okay, so with this question, you can find out what will be the number of cells after time t. Clear?
Sir, is N dependent on T, or are these two things independent?
Yes, obviously, n will be dependent on t.
Okay. So, sir, is there any expression to find N in terms of T or anything?
You go that you see the equation 2 by designing generation time
Okay. Got it, sir. Got it.
as g and total time as t. The equation 1 is simply, it has been written as Nt is equal to N0 into 2 to the power t by g. Okay, and this mu is taken as a 1 by g, which defines as the specific growth rate inserted into 2. That is Nt is equal to N0 into 2t mu, and g is the generation time, small g. It is the generation time means mean doubling time. So in generally, let us say g is 20, g is 20 minutes for a particular bacteria. So after every, if initially there are 4 cells, so after 20 minutes, it will become 8 cells. After 40 minutes, it will become 16 cell. Would you get? So after every 20 minutes, it will go on increasing like that, doubling like that, whatever it was. So that's so now equation 3 can be nt is equal to n0 into 2 to power t into mu. Mu is the growth rate. Okay? And after taking the logarithm of the equation 3, if the 3 is plotted on a semi-logarithmic diagram, you will have a straight line, which means that during exponential growth, you obtain a straight line like a log nt is equal to log n0 plus t mu log 2, which is shown in this figure, logarithmic phase that you see. These are the very simple mathematical equation. That's first log t is equal to log n0 plus t mu log 2. So with this, you can now. And I already explained the balanced and unbalanced growth. The growth culture is related to the composition of the medium. If all the essential components are available, the growth is balanced. However, if one or the several essential components are missing, the growth will become unbalanced. Growth will be terminated, and which often leads to death of the culture. And that again, the nature has provided some protection. Otherwise, if you see that a single bacterial cell, which is that even minus that is the cell weight, even micro weight like that, very minute. So after, if it is there for 40, it has a 20-minute generation time, and after 48 hours of exponential growth, it will produce a mass which will be 4,000 times more than the mass of this earth. So you could imagine what will be the situation. So this growth that nature has provided some sort of protection, like after some time, either the energy resources are depleted, okay, or the growth medium nutrients are depleted, or when the microorganisms are growing, they are producing certain metabolites which are toxic metabolites. So there may be accumulation of toxic metabolites, and eventually the growth ceases. So if you see that in the figure here, the graph, you can see the cursor, someone can affect, someone can confirm, are you able to see the cursor in the graph? Okay.
No, sir.
Sir.
Are you not able to see the cursor in the graph?
No, sir.
Okay.
So can you use a laser pointer or something like that?
That in fact I don't have. That is the problem sometimes.
Okay. Okay.
Laser pointer again with the screen, it doesn't go. But anyway, you can read that in the here x-axis is time and y-axis is the number of cell. All right, log number of cell. Can you see the figure? Figure you can
Yes,
see.
sir. Yes, sir. Yes, sir.
X-axis and
Figure we can
y-axis
see.
you can see. All right. So what
Can
is
you use
shown
mouse
here?
point? See,
I was trying to show the mouse, but you are telling you are not able to see. The time, it is time in the cursor. Can you see the cursor in the time? No. But
no.
anyway, you can just, I will speak and you concentrate on that. This is figure shows that different life cycle stages of a bacterial growth. All right. So when you bring in a culture some bacteria which was, you can take the example of that is in our homes, curd is made or yogurt is made in the industry. So what they use for making curd into yogurt, they use some culture, bacterial culture in our homes, that the previous day curd is used for as a inoculum. All right. So that is what is that? That is nothing but some bacteria in the form of lactic acid bacteria, streptococcus lactis, etc. So when a bacterial culture, when it is brought into a new medium, So it takes some time period of adjustment, okay, to adapt the new culture, new time, new culture, new environment. And that is also some, depending upon how long it has been in the earlier culture, how active the culture is which you have inoculated into the medium. It may take some period of adjustment, and that is known as, during which there is no growth, and that is known as lag phase, lag, lag phase. You see the stationary part of the line, red line, initial, please.
Yes, sir. We can.
Okay, and then after that, abruptly you see there is a increase, exponential growth. That is here the log phase, exponential phase. Okay, that is straight line you are getting. That is the number of cells on y-axis, log number of cells on y-axis, and time on the x-axis. So after a initial period of adjustment, you are getting a logarithmic growth. There is a, that is a linearly, that is with the time it increases cell number. All right, so it is in the, and during this period of log phase, the bacteria is in the balanced growth. All right, so population increases rapidly until, so this will go to some level, that here you can say that it, a point comes, because this bacteria cannot remain forever in a log phase. So either there is a cellular reserve depletes, nutritional reserve in the media is exhausted, or in the cell some toxic metabolites etc. are there. So this growth ceases. There is a little period of unbalanced growth, and then the cell enters into the stationary phase. Stationary phase means that the population stage is stable. There is no decrease or increase in the population, and at this phase, organism produces 2 to the power 0 metabolites. Means growth is not there at all. Growth completely stops. Right? And after, obviously, that it cannot remain forever in the stationary phase. So after certain time, the cells starts dying, and then it goes to the death phase. So these are the 4 phases: log phase, lag phase, log phase, stationary phase, and death phase in a microbial growth cycle. In the period of this unbalanced growth, that is in the transition between log growth and stationary phase and log phase, that is the cells are in a unbalanced, means the cellular population. There may be uneven cell, uneven in number, uneven in their size. But if you take a cell in the middle of the exponential growth, it will be all of the same size, composition, etc. In the stationary phase, it will be all in the same. Maybe mostly the spores are there in the stationary phase. Okay. When you put in the food in a microorganism, so bacteria goes into the stationary phase. It is live there, but it doesn't grow and multiply. Finally, I think I will stop it here today, and next we will take up the what are the various factors, how they influence this growth, etc. in the next class tomorrow. Tomorrow we will meet again at 10 a.m. Tomorrow is 11 a.m. I think. Tomorrow class.
Yes, sir. 11 to 12.
11 to 12.
Yes, sir.
So if anyone has any question, we can take 1 or 2 questions. If you
Sir, like the bacteria itself produces secondary metabolites in a stationary phase, which in turn kills the bacterial population. मतलब खुद ही को मार देते हैं.
yes, you see that inside the cell, that is there may be toxic metabolites, accumulation of the toxic metabolites. Okay, degeneration, cells degenerate.
Okay.
And their content increases, and after they can resist. What happens in our, even our body also, after aging, after a certain time when we age, what cells degenerate, and to some extent the body is able to withstand it, resist it, and then after a certain stage it starts degenerating and it collapses. So similar, same thing there also it happens. Right?
Okay, sir. So suppose, sir, it is in its stationary phase, and now I supply more biomass to the bacterial population. Then, sir, what will happen? It will remain in the stationary phase, or it will again go to the log phase?
You see that stationary phase, you have to, there are bacteria has become dormant. So that will depend upon the what are the conditions. Suppose stationary, it has gone into stationary phase, but if you give a nutrient as well as the environment for its growth, it will grow and multiply. Like in the refrigerator, you have kept the food. All right. So
Yes, sir.
bacteria has become gone into stationary phase. But if you take out the food from out of refrigerator, keep in the room atmosphere, then bacteria will take again little period of adjustment, and then it will start growing and multiplying.
Okay, sir. So it requires biomass as well as necessary conditions for growth both.
Conditions. Yes,
Okay.
both. It will require both. And anyone else? Any other query? If no, then we will stop. Okay, bye-bye. We'll meet tomorrow. Till then, namaste.
Thank you, sir.